Okay, thank you very much, Vijay. I feel like this is one of these episodic soap operas you see on TV because as you remember our story, okay, I was just telling you that enhancers evolve, and I had just mentioned that the enhancer that controls stripe 2 from melanogaster, Yacuba erecta, and pseudo-obscura, when you express them in melanogaster, they make stripes. Now, there's an even more extreme case, came out of Mike Eisen's lab almost 10 years ago, that is another problem that's worth obsessing over, okay? Sometimes I see these problems, it takes years to solve them, but they really bother me, so I keep thinking about them. And this is another one, okay? This is a Drosophila melanogaster. This is sepsis synapsia. Okay, it is not only not a Drosophila, that's the genus, but it is not a Drosophilid. Okay, and it's not from the family Drosophilidae, it is from the family Sepsidae. Okay, so this is really fairly distantly related. Okay, I don't have a million-year figure for you in my head, but they're pretty diverged, okay? And you can see how diverged they are at the sequence level. I checked the paper. I think each black line is about 12 bases homology, but I'd have to check. Heredal 2008. And so these are two sepsid species, or even skip sepsis synapsia and Thamyra putris. Here's melanogaster. And you can see that the coding region is very highly conserved. Okay, so just think back to Hamad's talk. There is obviously strong selection operating on the protein sequence. And, you know, it hasn't changed much. But then, if you look off into the control region, you see that the sequence divergence is fairly large, even between the two sepsids. And from the two sepsid flies to Drosophila, okay, there's almost no homology. Now, there's been some dispute in the literature, but I can pretty much sum up to you that all of the homologies that you do see between the control region of melanogaster and the sepsid flies are essentially no longer than two overlapping binding sites. In other words, if you're looking at it at the sequence pattern, at the sequence level, the genes are really quite diverged. And I'll show you a quantitative map of that. Oh, wait, I can move slides through here if they don't mess up. Okay, but here's the thing that is extremely puzzling. If you take these highly diverged stripe 2 enhancers that have no sequence homology with the melanogaster enhancer, and you put them in melanogaster embryos, they express a stripe 2. Okay, it turns out if you measure carefully, this stripe looks like it's a little posterior of the Drosophila stripe 2. Okay, so this is another experiment worth some obsessive attention. Okay, because here you have concentration of function, if you call function making a stripe, but you don't have conservation of sequence. Okay, so something is conserved, okay, but what is it? Well, and I just want to quantitate that comment about sequence divergence. So one way of measuring the divergence of sequence is the so-called Levenstein or minimal edit distance, okay? And this is the number of single base changes that you need to make to transform one sequence into the other. So in other words, every substitution of one base, that counts as 1. If you have insertions or deletions, every time you add or remove a base, that counts as a distance of 1. Okay, so what you're seeing here is the black is just the length of the enhancer. but the red is a measure of the distance from melanogaster. Okay, so you can see that erecta and jacuba are diverged, but they're reasonably close. Pseudo-obscura is further away, but by the time you get to Thamyra putris and sepsis synipsia, okay, this is a lot of edits. Okay, you have to change about 1,000 bases. Okay, so I wasn't kidding you when I showed you the last slide that showed the near absent sequence homology. So when Aram Kim was working with this model, I basically told him to get his hands on every enhancer that he could find sequence of in the literature and just plug it into the fit that he did on those fusions. Okay, so remember all the way back before lunch, I was showing you a fit to data from the very peculiar results where you took two enhancers and you put them right next to each other, and the result was you got a completely new pattern. And that was just done within Drosophila melanogaster. Okay, so it turned out this was very successful in predicting things. Now, what I'm showing here, technically, I would call retrodictions. Okay, so a retrodiction is when your model gives you something that you didn't put into it, but somebody has already measured it before. Okay, and I will show you a genuine prediction down the road. For example, these are various Drosophila species. Okay, they're not perfect, but for example, here's pseudo-obscura. Okay, stripes 2, 3, 4, 5, 6, and 7 of melanogaster, and the expression of pseudo-obscura is correctly predicted. Now, what's more radical is the model predicts the location of the sepsid stripe in melanogaster. Okay, notice that it's a little bit posterior to the native stripe 2, but in the paper by Herodal, that in fact was what they reported, was that it was a little bit posterior to the native stripe 2. And so, you know, this is sort of very gratifying. Now, the point is, though, we don't, if you're an engineer, you really care about predictions, because if you build a bridge, you predict that it won't collapse, and you had better be right. Okay? Now, everybody likes predictions because, well, basically, one's colleagues, you get more credibility from your colleagues, and your grant is more likely to be renewed. So there's a certain sense in which predictions are valuable sociologically because they make people believe you. Okay, but our idea here is not to tell fortunes. Okay, the whole point of doing this is to understand what's going on. And so one issue that goes throughout the work that I do is it's great if you can predict or retrodict something. Okay, but the point is what you really want to do is understand the system. Okay, and the prediction is just a means to the end. If you had a perfect computer model that accurately predicted everything, and it was super complicated, that really doesn't get you anything because you don't understand the model any more than you understand the embryo. And this is always a tricky thing with these models that have a lot of parameters and stuff. Okay, so the question is, why is it that the prediction is correct? Okay, and so one of the reasons these big promoters are so extremely confusing is that they have so many binding sites. Okay, so, excuse me. So what you're looking at here is you're looking at the binding sites around the minimal stripe element for stripe 2. Now, I should point out, and it's shown in a later figure, not so much on this one, that I said this is the minimal stripe element for stripe 2. The whole enhancer is about twice as big. Okay, and so this was classic work by Mike Levine, making it as small as possible. And I'll come back to that in a minute It turns out when you look quantitatively, the whole enhancer expresses about 5 times more strongly than the minimal enhancer. Are we getting echoes? Maybe I'm having auditory hallucinations. Okay, so the problem then is you have this model that's reasonably complex, and you want to look inside it and figure out what's going on. So when I showed the model, I said something about adding things up to get activation, and the response of the model looks like a sigmoid, okay? And so the idea is that this was a diffusion-limited Arrhenius mechanism. So essentially, this is going to be delta A. This is going to be delta A. Actually, I drew it with the wrong sign, because if you lower delta A, you get more expression. And so essentially what you're looking for, you ask yourself, how do you get the stripe from the binding sites? What you're basically looking for is you want to lower that activation energy barrier. And so if this is delta A, you're lowering it by delta A. Okay, and I have that on the board, so it will be important for what's coming. And everybody is more tired now because we're not bright-eyed and bushy-tailed. Hey, by the way, the posters were great. I want to thank everyone I talked to at the poster session. So interrupt me with questions. So here's the problem. There's all these binding sites, but the thing that you have to remember is at every point in the embryo, there are different concentrations of transcription factors. Okay, so the thing is that not all these binding sites are active in doing everything. So the trick is you have to fix a location in the embryo. Okay, in this case, we want to fix the location to be stripe 2, and then you essentially want to say, where is the activation coming from? And so you have this very confusing set of binding sites. The activators are up here, the repressors are down here, and you just look and say, what could be going on? You take the top 80% contributors to the gene expression, okay, and then since all the regulator sites are acting over a short distance, yes? Don't worry about a microphone, but yell it out louder, please. No, just louder.
Yes, Professor, I would like to know, how do the binding site affect the regulatory mechanism of the activation, affect the activation mechanism?
Well, at the level of phenomenology that we're doing, we just assume they all contribute to delta A if they have stuff bound to them. You know, at a chemical level, there's more to it than that. Is that the answer you were looking for? Yeah, they stick to, well, they stick to the polymerase, whatever that means. Right, and so in other words, for all the activator sites that are contributing to this lowering of delta A, there will be other regulator sites that are interacting with them, maybe through cooperativity, maybe through repression, or maybe they're co-activators, and they're the things that are making that activator active. Okay, and then it could be that some sites are almost completely repressed. Okay, so this capital F is the fractional occupancy after you've corrected for short-range repression. So I won't torture you by going back to the slide that shows the full model equations, Okay, but the point was that that equation that I explained with the big product was essentially how you get capital F from the uncorrected activators. And so you throw away anyone that isn't active, and the result is that suddenly everything looks much more understandable, because most of these binding sites aren't doing anything. Okay, if you're at the anterior border of stripe 2, then the active sites are reduced to about 24. Okay, this was the thing that was missing up here. These two conserved blocks are the classical boundaries of the full stripe 2 enhancer, and that will be important in a little while. Okay, so now suddenly things look a lot simpler. Okay, and it turns out we can go a little further. Okay, and this was actually a bit of a surprise, because more than 80% of the activation is coming from exactly 2 binding sites, and they both bind hunchback. This is a named site. It was found it was described in the classic paper by Small and Levine, hunchback 3, and this one here is hunchback 2, and they're each surrounded by interacting binding sites. And so suddenly we're not looking at a great zoo of binding sites all over the promoter. We're looking at 2 very specific clusters. Okay, and then when we look down and ask, okay, what do those contribute to activation? Okay, what you're seeing below here, okay, is on the position on the embryo as usual on the horizontal axis, and the delta delta A on the vertical axis. Okay, in other words, the vertical axis is showing you how much you bring back down the activation energy barrier and how far up on this exponential sensitivity you are. Okay, so what you see here is the hunchback 3 cluster. Okay, this black is the total activation provided by the enhancer. Okay, and this activation back around stripe 7 is not an accident. I'm not going to discuss it in more detail here. Okay, and so in black is the total activation. In red is the activation that's provided by this particular cluster. So you see that the hunchback 3 cluster forms actually a stripy pattern of activation. The hunchback 2 cluster essentially boosts things with only providing a minimal amount of stripe contribution. Okay, and when I point out to you that this whole enhancer between these 2 blocks actually turns out to drive expression 5 times stronger than the expression of the minimal enhancer in red, okay, you can see that really the minimal enhancer is essentially driving from the hunchback 3 cluster, and the full enhancer has this extra kick from the hunchback 2 cluster. And this is one of the reasons why the study of these enhancers is so puzzling if you do experiments, because it's full of situations where a piece of DNA that does nothing at all by itself, if you add it on to another piece of DNA, it modifies its function, or the 2 together have a new function that wasn't present before. Okay, and this is the kind of thing that models are very useful for reading off. Now, I might add very briefly, and one of the reasons that we had to do some other experiments, we're about to, this is still unpublished, is that, you know, it said that extraordinary claims require extraordinary evidence. So one problem here is the classical story of stripe 2 that you can look up in textbooks actually is that the main activator is bicoid, not hunchback. Okay, but if you think about it a little, you think about it a little bit, that's not so crazy, okay, because these bicoid sites are co-activating hunchback and turning it into an activator. And furthermore, The other reason that makes sense is that in the fusion experiment, okay, you were bringing stripe 2 and stripe 3 together. Okay, stripe 3 is known to be repressed by hunchback, and the stripe 3 enhancer is jam-packed with hunchback binding sites. So once you bring them together, the hunchback sites in the stripe 3 enhancer are within co-activation range of all the bicoid binding sites on the stripe 2 enhancer, and that's what boosts the expression so strongly in the fusion. Okay, so all the stuff I've been telling you is that essentially the hunchback 3 cluster codes for each stripe 2 expression. It essentially is the functional component of MSE2, and the hunchback 2 cluster codes for amplification of expression, but does nothing by itself. Now, I'm going to say something that's very speculative here, okay? Because these are the 2 functional clusters we've found so far, okay? We need to find more, and we have more places to look, lots more places to look, but it does suggest an analogy for the genetic code for regulation. Okay, I said that the genetic code for proteins is like an alphabetic Western language. But the genetic code for enhancers, if you believe the story I just told you, is a little bit like Chinese characters, okay? Chinese characters very frequently have these 2 components that are called radicals. In this case, this is the character for flies. This character means insects, worms. And this radical is simply a modifier. It's not used alone now. It has an archaic meaning. I must tell you, I don't know Chinese. My student, Zhihao Lu, helped me with this. I did know about radicals. And so what does that suggest about what's conserved, okay, across these various species? Well, what you have to do is look at the cluster structure. Okay, so it turns out you can actually make sense of this if you look at the sequences of these various species. Okay, so what you're seeing on the right is, what you're seeing on the right here is the enhancers from Melanogaster, Yacuba, Erecta, Pseudoobscura, Synipsia, and Putris. And among the Drosophilids, you can, although the sequences are diverged, you can trace individual binding sites. And there's essentially a compensatory evolution situation operating here. These named binding sites can be traced through the different species, essentially because the sites connected by dashed lines are closer to their cognates in other species than they are to other sites in the same species. And so what you see here is that although they're, got to press the right button here, although you can see that the detailed arrangements of the sites are preserved, the general overall structure of the cluster is maintained. And if you look very carefully, I won't go through the details, what happens is sometimes a site is lost and a new site comes into being. Okay, so this is telling you what's conserved among the Drosophila species. Now, what about the sepsids? Okay, here you see something more strange and unexpected, okay, which is in Synipsia, you start to see sites for another gene that I haven't said much about called Caudal. Okay, it's present maternally and zygotically. And for a long time, its function was kind of obscure, but essentially it was generally thought to be patterning the posterior part of the embryo, because that's where its expression is highest. And so what you see in Synipsia, and very strongly in Putris, is you still have these hunchback sites. okay, just like you have in melanogaster, but they're no longer being co-activated by bicoid. They're being co-activated by caudal. Okay, so this is another situation of extraordinary claims requiring extraordinary evidence, because if you were to grab sort of some random Drosophila developmental geneticist in the street and say, oh, I just discovered that caudal is involved in activating stripe 2, they would say, well, you're crazy, okay, because everyone knows that stripe 2 is on the wrong end of the embryo for that. So this made us very nervous, and I just actually wanted to publish the paper, but my student Aram Kim, I don't know, there's an famous American story by Mark Twain about Tom Sawyer. And it has this, there's one episode in it where Tom Sawyer is supposed to paint a fence, and he doesn't want to. So he just tells all its friends that painting a fence is really fun. And eventually they all paint it for him, okay? So Aram was going to leave the lab, but he persuaded my other student, Pongyao Jiang, to do the experiment. And she was new to Drosophila. She didn't know how hard this experiment was. Okay, it was insanely hard. I won't go into the experimental details, but I'll just mention that caudal is a gene that is required both zygotically and maternally. Okay, so immediately you might start scratching your head and say, well, how can you have a stock in the lab? Okay, because you have to have mutant embryos from mutant mothers, but the mutant mothers, of course, will be dead. So how do you get those embryos? Well, you do it by a trick, okay? And the trick is you can make mothers that have patches of tissue that are caudal minus, and as long as those patches of tissue include the ovary, then the whole thing will work, okay? It's pretty hard, but I won't get into the details of the crossing scheme. I'll simply say that as you would expect from the diagram I showed, what our model predicts is that in the absence of caudal, okay, the putris, which was the one clone that was still around in Mike Eisen's lab, would still be expressible in the presence of in a bicoid mutant, but would be absent in a caudal mutant. And so Pongyao tested that. Okay, so this is an actual prediction. It was really very, very hard to do this experiment because the report, it was very hard for certain technical reasons to do the experiment. You'll notice these embryos are not stained fluorescently, and that was part of the difficulty because the putris reporter is a CFP reporter, not a LAC-Z reporter. But you can see in wild type, you have the melanogaster stripe 2, the putris stripe 2, but here in embryos that are missing maternal and zygotic caudal, in fact, the melanogaster stripe is reduced, but the putris stripe is gone, okay? And so in this case, this is an actual prediction, and it was correct. Okay, so this is is telling you the story of how, although the sequence is not preserved, these functional clusters are preserved. And so, you know, this has certain implications for the genetic code. And maybe if we can find more clusters, and actually need some new students because a bunch have just graduated. It may be that this idea can be generalized. Okay, don't completely believe me without evidence, but it's an exciting possibility. And so, in this analogy to Chinese, I found as many Chinese fonts as I could. Now, the rest, I somehow thought that with an hour and a half, I was going to go through a lot of stuff. Okay, so there is another part to this talk, and I'm going to skip most of it. Okay, I just want to make one or two comments about the high points. Yes, Kavita. No, what I mean by yes. Okay, this is what I mean by canalization in cis, because you change the sequence a whole bunch in cis, but you didn't change the expression. Okay, and these instances of canalization across species are quite fascinating. So there's a paper by a guy named Rendell. It's another one of these ancient classics, like Waddington's paper. And so there are 4 scutellar bristles, essentially, on the back of the fly. But you can create a genotypic situation where the number of bristles can be variable, and occasionally you get an extra bristle. So if you take the flies that have an extra bristle and you cross them with other flies that have an extra bristle, you can get the presence of the extra bristle to be more and more stable. Okay? However, it's not like you can get as many extra bristles as you want. You can essentially get a maximum of 6. And I forget which species it is. That's found in another species. Okay? So there's sort of this idea that in ways that are kind of obscure, okay, this and this is another example from the data that I've shown, that this canalization phenomenon extends through evolution in ways that are really poorly understood. So like I say, everything I'm saying about canalization just scratches the surface, okay? And partly I'm telling you about this because maybe, you know, some of you will decide to work on this and have something to contribute. Okay, so I just want to say 1 or 2 things about the whole locus. It was thought for a long time that enhancers were additive, okay? They're not. And there's various ways that used to be sort of a heretical position. Actually, everybody agrees now that that's not the case. I won't go into things like, I say, for example, it is well known among experimental biologists now that there exist things called shadow enhancers. So if you look very carefully on many, many genes, Eve is not an example, but if you look at the enhancers that drive a particular expression domain, if you look carefully, it often turns out that there's more than one that drive the same domain. And furthermore, if you have both enhancers present, you have significantly less noise and less fluctuations than if you only have one. Okay? And I should say that there's been a previous paper published on the whole locus, Sami and Shinha. Okay? They have a beautiful model, but they assume the existence of enhancers as a condition of the model. Okay? We did not want to do that. We wanted a completely general model where the existence of enhancers emerged from a more general idea. Okay? And so the idea is, of course, you know, I've been talking about the range of these interactions. Okay, well, range is along the DNA. Of course, the transcription factors aren't sending messages on their cell phones. You know, we believe that the DNA folds back, and these enhancers are in contact with the basal promoter. Okay, so the basic idea here is simply that only some of the cis-regulatory DNA can be in contact with the basal promoter at once, and furthermore, its chances of being in contact are proportional to how much activation is coming from there. Okay, so it's sort of, a lot of you are graduate students. It's like if you work in a lab. Okay, you may not be competing with the other students, but it's likely to turn out that if you're getting the most results, you're going to spend more time talking to your advisor than the students that are getting fewer results. So I'm just going to, this is published now. It works. Okay, I'm going fast. This is basically showing, I'm really going to go fast through here. Okay. This is essentially showing that it can give the expression of individual enhancers correctly, okay? And we also show And so, actually, I'm going to skip through this. You can take the system apart and see what genes control the borders. But again, if you're interested in this, read the paper. I just want to make the point, 2 points here, okay? One is that although we did not put in the presence of enhancers as a assumption of the model, simply limiting the amount of DNA that can be in contact with the basal complex, okay, is enough to give you a presence of enhancers. And in fact, we thought that we had predicted a new enhancer. So, you know, I just sort of happily showed you a prediction that was actually correct. There was a prediction that wasn't correct, and it was on, well. So this graph shows distance along the embryo on the vertical axis, and it shows location along the gene on the horizontal axis. And this heat map shows where the activation is coming from. So here's the stripe 5 enhancer, stripe 4 and 6 enhancer, stripe 2 enhancer, see that's up by stripe 2. So we were very excited to see this, okay, because we thought that we had identified a new stripe 7 enhancer. Okay, so look, I have to be honest, not all predictions are right. Okay, I'm not going to show you, if you find me later, I can show you the embryo. It gave a totally crazy pattern. And so people are always saying, well, what about chromatin? What about chromatin? You know, you guys don't count chromatin in your model. Well, really, for a single enhancer, it's not very important. But in this case, actually, it is important because there's data out there. Wait a minute. Here we go. There's data out there from Mark Biggin's lab that has mapped where the heterochromatin, the inaccessible chromatin, is in the Eve locus, okay? And essentially, it turns out, okay, that this spurious enhancer we found is actually in an inaccessible portion of the chromatin, okay? And then basically, if you add chromatin structure, we can predict the enhancers correctly. Okay, so that was part 1, extended. Okay, see, I go faster in the afternoon because everybody's tired. Okay, so Sanjay asked about, you know, what about things that happen in time? Okay, so the second part of the talk is about things that happen in time. And before I get into the second part of the talk in detail, I want to show you a beautiful piece of data that is not from my lab. Okay, it involves some experimental techniques that we want to start using. Okay, this is data from Michael Levine's lab, and he very kindly gave me the movie that I'm about to show you. So this is just a piece of beautiful experimental work, and what you're going to see, let me find it here. An X term, that's not what we want. And how do I get this on the whole screen? No. Where is the, I want to, green, oh, okay. Here, I'll get it. These great. Okay, so let me tell you what this is. This is a movie, oh, there you go. I'll show it twice. We'll see it again, don't worry. Okay, it's starting to gastrulate. Okay, so I'm going to show it again. Okay, this is a live embryo, and it has been altered with CRISPR-Cas9. I think by Perry in Mike Levine's lab, so that you can see the transcript initiation. Okay, this is the Fushitaratsu or FUTS gene. And so what you're going to see is the stochastics of transcription initiation. Okay, I don't have a model that describes this in full detail. Oh, there we go. Okay, those are the synchronous nuclear divisions. There, those are the nascent transcripts. Now watch, it's going to make stripes. 7 stripes. Okay, now it's gastrulating. Okay, I couldn't, it's not even my data, I just couldn't resist showing you this. Okay, but yes. Excuse me? The stochasticity is that these little, I'll run it one more time. Okay, see these little dots here? Those are nascent transcripts that are just beginning to be expressed. Okay, so essentially every time you see a white dot, that's the initiation of transcription. Okay, so it has been shown that actually the initiation of transcription is a stochastic phenomenon. And you can say things about it with simple models that essentially give you probabilities of it flipping between on and off. Okay, but the real complicated thing is you would really like to know how all the transcription factors that I talked about actually control this process. So I'll tell you just very honestly that at the moment I have no clue. I have absolutely zero clue how to set up a model that would give me the information that I would like to have. Okay, there's things you can do with much simpler models with 2 random variables, one for the transcription initiation and one for the number of protein molecules, but that does not include the genetic control. So we'll see, you want to see it one more time? It's pretty short. Okay, watch those little white sparkles. And by the way, there's those 13 synchronous nuclear divisions. You can see them too. There. The transcription shuts off during the division. We finished the 13th division, and now you can see the formation of stripes. Okay, so this is more canalization, and I will still, this is going to spill over a little bit to tomorrow afternoon, but I won't let it spill over so that it gets in the way of these certain Boolean models that I want to talk about tomorrow. Okay, so look, I like to go from transcription to pattern formation. I mean, that provoked some questions from Sanjay and 1 or 2 other people because you were wondering about what the whole system is like. Okay, so I just moved that to this slide. Okay, there are 14 genes that are required for making the segmental pattern. The patterns of all of them are shown here. Okay, this now seems hopelessly old-fashioned and obsolete after the movie. Okay, this is simply to make the point that the patterns change in time. Okay, what we have been working with is reassembling the time course from fixed tissue. Okay, and it's great to make the movies. For stochastic stuff, you actually absolutely need the live movies. There are, however, advantages to the fixed tissue also. Okay, so I want to remind a little bit about, I brought in canalization in cis, okay, that was within one gene. Okay, now I'm going to be talking about canalization in trans, and that's across the whole segmentation network and a little bit more generally in the organism as a whole. Because remember, I talked about this idea of the buffering of phenotype with respect to genotype. So now we go back to Waddington's papers. Okay, and I quoted that paragraph. The quote is coming again. I won't go through it in detail. But one of the remarkable things is how Waddington made that made that deduction. Okay, first of all, he noticed that tissue types are discrete. They're not a continuous mix. Okay, this may seem obvious, but actually this is kind of a profound thing. Somehow you have to generate discrete tissue types. The other thing that Waddington noticed, which actually any experimental geneticist knows, it's just kind of a fact of life if you work at the bench, that the phenotype of animals, although it's very constant in wild type, this buffering of phenotype by genotype is completely lost in mutants. Okay, and this is an example from an old paper by Eric Wishaus, who first isolated the segmentation genes. This is a normal larva. Okay, I keep talking about segments. I forgot to put the picture in of an adult fly, but in this larva, you can see the 8 abdominal segments. And this is a Krupel mutant. Okay, so you see why it's called a gap gene. There's a lot of missing segments, and some of them are mirror image duplicated, but this phenotype is variable. Okay, now I've already given you the Waddington quote with the beautiful picture of the canyon, so I won't spend time on this. But I do want to say some things about, I did claim that this Waddington paper was enormously fertile, intellectually fertile. It's actually had a very, very remarkable, there's this whole idea of, I say regulation of morphogenetic fields. So morphogenetic field is just a collection of cells that starts out uniformly and becomes differentiated into some kind of structure, or becomes determined to form a certain structure. So the blastoderm is a morphogenetic field. Early in the blastoderm, each cell doesn't know what it's going to do. By the end, by the time gastrulation starts, each one has a stably determined fate. Okay, now this was something that was discovered by the great titans of embryology late in the 19th century. And there was one guy named Hans Driesch who did some experiments that were really quite remarkable. He showed that you could take a sea urchin embryo in the 4-cell stage, cut it into 4 pieces, and each piece did not develop into a quarter sea urchin. It developed into a whole sea urchin, okay? And Driesch, who correctly concluded that the chemistry and physics and mathematics that was available at that time could not possibly explain this phenomenon. So he stopped doing experiments and became a vitalist. And I don't know if you know what vitalism is. It's the old idea that living systems have some special spark. Okay, I mean, they do have a spark, but I would say it's an emergent property of molecular interactions. It's not some peculiar golden liquid. Okay, so he became a vitalist. Okay, now, this business of tissue types being discrete, okay, it's not that Waddington was the only person who noticed this. Okay, way back in 1940, Leo Szilard and Hans Delbruck proposed that cell types can be described by multi-stable dynamics. Okay, I mentioned Waddington, and one remarkable outgrowth of this was there's a whole field of mathematics called topological dynamics that was invented by Rene Thom, the Fields Medal winner. And as Thom says in one of his books, he thought of all this up in order to put the creodes, Waddington called these things creodes, to put the creodes of Waddington on a rigorous math basis. Okay, now there were 2 problems with what Thom did. Okay, one problem was that he worked out the math only for potential systems. You guys know, especially the students, you know what a potential system is relative to an autonomous system relative to a non-autonomous system? I see blank looks. Okay, I'm going to take a very brief digression, if you'll forgive me, because this is a class. Okay, suppose you have a system of ODEs. It could be a very large system of ODEs. Don't even think about PDEs. They require things like existence theorems and so forth. So, you know, we're thinking about something in the lines x dot. Well, x dot depends on something. Okay, so the simplest situation is that x dot is the gradient of some stable potential. Okay, that's called a gradient system. This is called an autonomous system. And you might have something that had explicit time dependence. This is just a non-autonomous system. And of course, really, everything is much worse. So, of course, really in a biological system, there's all kinds of stuff left over from things that happened in the past. You're going to deal with a system with history, okay? These are really a mess. You want to avoid working with them whenever possible, okay? There's a beautiful theory, not the theory that was invented by Tom, but all the things that Tom did were extended to autonomous systems within about a decade, by the end of the 1970s. And there's a beautiful theory of the qualitative properties of these systems. And I'll say some things about that later. At minimum, it turns out you have to treat the fly embryo as a non-autonomous system. There are some things that are time-dependent inputs, essentially gap genes that are put in by the mother, but you can sort of get around that problem because all the good theory about autonomous systems has to do with things that happen at infinite time. And of course, biological systems never get to infinite time, so you can just, there's various ways to get around that problem. Okay, now look, I get to give my little editorial comments here. Okay, so I do want to say something about landscapes. Okay, and I can address this to the physicists in the room who will understand exactly what I mean. Okay, some people have an idea with Waddington, they think about these landscapes. And it's very easy to talk about landscapes, you know, fitness landscapes, whatever. Okay, those are all potential systems. Okay, but as is implicit in a lot of talks that we heard, okay, for example, the one by Hamid Malik, okay, and also, in fact, an excellent poster that I saw, Eva? Anyway, some great posters out there where you represent fitness by a potential, but it suddenly changes, okay? That this potential surface is never the same. So if you're a physicist, okay, this makes perfect sense. Okay, if you're trying to do electromagnetic theory, you can't do it with just a scalar potential. That's just going to give you electrostatics. And in general, if you want to talk about things that happen in biological systems, there's only so far that you can get with a picture with landscapes. Okay, it's very good as a metaphor. It's not even so great as a data visualization tool because, of course, you can draw a picture of a landscape, but it only helps you if it has 2 independent variables. If it has 30 independent variables, then that picture is not really very comprehensible. Okay, so I just wanted to work that in. In any case, the real problem with Tom's stuff is there was no data, but now there's data. Okay? So I have to say a few things about some classic ideas from developmental biology. There is a, PJ. Oh yeah, maybe.
You want to look at why there are discrete and different types of tissues and
Yes.
and how that's connected to patterns that come up in
Yes.
okay. So first of all, even if I don't have patterns, I could have different kinds of tissues, right? I mean, this could be a more cell internal process.
Well, what do you mean if you don't have patterns? They usually make patterns.
They usually do make.
Always.
No, but
I mean, for example,
sure.
X inactivation
Sure.
in mammals is not much of a...
Yeah. No, I'm trying to distinguish what is something which requires external signaling versus something which is cell autonomous.
Well, essentially, the idea is that all of these systems have parameters associated with them. And so the signaling can be thought of as a change in the parameter. Okay, so for example, the stuff I'm talking about doesn't really include signaling in a classic sense, but if I'm talking to a group of mathematicians, let's see, do we have some bicoid here? There's bicoid up here in red. You can hardly see it. But I tell them, okay, you see this pattern, this is a parameter. Okay, and in stuff I'm going to show you later, bicoid will be a parameter. I don't know if I answered your, did that vaguely approximate an answer to your question? Okay. So I want to contrast the, yes. Please, please. Just say it loud so everyone can hear.
I mean, I think the way it helps to think about this is that each cell may internally run an autonomous system, but that cell will also excrete molecules that can diffuse around and go to neighboring cells and can impinge on it and act as an external source on those neighboring cells. So you have these two sort of things happening in parallel, because on the one hand, you may sort of think each cell is sort of has an internal program that's a function of just the internal concentrations of all the things that it has, but they communicate in space with each other, and then often you have to model that as an external field that impinges on your cells. You agree with that, John?
Does it require signaling, or can you have it just by cell autonomous process? That's not.
You could have it.
You could have it.
Yes, you could.
Okay.
You could. They tend to be more chaotic. Okay, so for example, in classic Turing systems with a pair with activators and diffusible inhibitors, they always form patterns, but the patterns tend to be quite variable. They can look like stripes or spots or fingerprints. And see, look, in a larger sense, if you're trying to figure out a particular system, which I now know from looking at the posters, actually, a lot of you know quite a bit about, the real trick is to use this math theory in a useful way to figure out something about your particular system. And that always involves a little creative play. So there's this classical idea from Louis Wolpert back in 1969, the idea of positional information and a morphogen gradient. Okay, and so the idea is that you organize tissues into different cell types by having some molecule that's present in a varying concentration across space. Okay, this is sometimes called the French flag model. That suits me because I work on the anterior-posterior axis. Okay, if I was working on the DV axis, I might prefer a Russian flag or a Dutch flag model. And so the idea is that the cells read off the concentration of this morphogen, and if they have high concentration, they follow the blue fate. If they middle concentration, they follow the white fate, and if they have a low concentration, they follow the red fate. And, you know, I suddenly realized that if I want to be careful to understand things, I want to just pause for a minute and say exactly what I mean by fate, okay? So there is a classic thing that was found by Spamon in the late 19th century, that before morphogenesis occurs, the cells somehow become internally determined to follow a particular developmental pathway. And so the type of experiments that you do, okay, that were done classically, is you do surgery on embryos. And you take a little piece of tissue that's in one place and you transplant it to a different place. And if it grows according to the place you put it, then it didn't sort of, it's not determined. Its future state is determined by its embryonic environment. And it turns out that early that's what happens. But after a certain amount of time passes when you do this transplantation, the cell grows not according to where you put it, but where it came from. In other words, there's something about its internal state that they say determined. Okay, and the math that I'll be getting into at the end of this talk or tomorrow, okay, is the sort of math that you need to use to describe this phenomena. Okay, so I have to say what fate means, because it's kind of it has meanings in general English also. Okay, now there is sort of a problem here, because I've said that there's a lot of embryonic error correction. Okay, you look at this French flag, and it doesn't show error correction. It does not canalize. If you perturb this concentration gradient just a little bit, then the fates will shift. Okay, and so in fact, people were very interested in finding these. Okay, so this is, bicoid is actually not a very typical morphogen. There are actually morphogens in vertebrates that are much more complex, and there's actually have some amazing properties that I won't get into, but bicoid was the very first morphogen found. Okay, and this is not the original observation, which was from Drever et al. 1988. This is from Gary Struhl's paper, but I'm showing it because, well, I'll tell you why I'm showing it in a minute. But the idea that the way you demonstrate something is a morphogen is that you want to rescale the gradient and show that the cell fates change accordingly. Okay, and so it turns out you can do that very easily with bicoid. Okay, it's been known for a long time that bicoid has an exponential profile. It's high in the anterior and low in the posterior. And you can rescale it by doing genetic manipulations where you change the number of copies of bicoid in the genome. Okay, and that essentially will linearly rescale the gradient. Okay, now, hunchback is a particularly simple case of response to bicoid because it is known that hunchback responds to bicoid, okay, also some other things. And so the idea is when bicoid is above a certain concentration, hunchback is on, and when it's below, hunchback is off, okay? And this is showing the fact that actually happens. 1 copy of bicoid, 2 copies, 3 copies, 4 copies. Okay, but there's some problems, okay? One is if you look quantitatively, it actually turns out these borders do not occur at the exact same concentration of bicoid. And there's another problem that's, yes? Excuse me? A morphogen, okay, I'm going to give you 2 answers because if you talk to some developmental biologist that you meet at a meeting, he'll say that a morphogen is this thing that Wolpert proposed that's present in a gradient. And I have to say that that is in modern times the general meaning. If you talk to biologists, that's what they mean by a morphogen. I can't help saying that actually Turing had a much more general definition of a morphogen that I actually like better, which is that a morphogen is any chemical that makes the pattern that will be followed in development later. Okay, so in that sense, all of these genes I've been talking about today, they're all morphogens. Okay, having said that, if you're talking to your friend, a biologist, he or she will definitely mean this. Okay, yes. Yeah, that's basically the idea. Hence the name. Okay. So now we get into some really weird stuff. So at the sort of dawn of quantitative investigations of this, there was a very unexpected paper from Bahram Houchmandzadeh that indicated some phenomena very similar to this. This is from our data. Bahram's results were very similar, and essentially what he looked at was the variation of bicoid gradients across individual embryos. And if hunchback is being triggered at a particular threshold of bicoid, you look across the population and the location of this hunchback border varies by about 5%. Okay, well, all right, it means that if it's being triggered from the bicoid threshold, you would expect 5% variation. If this is the average threshold, you would expect the location of this to vary by 5%. In our data, it varies by 25%. Now, I have to admit that the very best data on this problem was actually collected by Thomas Greger at Princeton on live embryos. We actually have, our data is pretty reliable, but bicoid is a very tough gene to standardize the expression level. So I believe Thomas's data, except that even in his data, there is a discrepancy, okay, because the spread of thresholds is about 10%, but the spread of the hunchback border is smaller, okay? So what's going on, okay? And so I want to give you, so we analyzed this with a model, okay? Now you're a bunch of theoreticians, so I'm actually going to sort of derive the model, okay? So really, all it is a Taylor series, okay? And actually, before I put up the next slide, I want to remind you okay, that in the transcription model, there was this delta A that lowered the Arrhenius barrier. And there was all this complicated garbage in the transcription model, okay, about coactivation and cooperativity and this and that. It all went into delta A, and we got the gene expression level out of that, okay? So what I'm going to show you very briefly, essentially you can simplify the transcription model greatly by simply taking that insanely complicated expression for delta A and expanding it in a Taylor series. This is some work I did with Vitaly Gursky. Okay, he likes slightly different notation, but basically this is the same stuff I showed you on the big slide. Okay, you basically find the occupancy of sites. This is the repression equation. And then what I've been calling delta A here is in this notation U of V. Okay, so now we just do a Taylor expansion of U of V to first order, okay, and you have to choose a starting point. This is an important point. It turns out it's not very sensitive to where you do the Taylor expansion. Okay, let's see. W. Where is W? Oh, thank you. Sorry, look, at least you're reading the slide. Sanjay, W is the transcription rate. That's the derivative of mRNA concentration with respect to time. Okay, I apologize for the fact that notation is different. I really should remake this slide. U of V is the diffusion limited Arrhenius rate law that gives you this sigmoid. Okay, equals W. Okay, so the idea is that in this threshold function, the things that you use to get the quantity on the horizontal axis involves an extremely complicated calculation that has all sorts of things in it. It has many binding sites. It has those big products. You would never want to write it down or take any of these derivatives except using Mathematica. But nevertheless, you can do a Taylor expansion. Okay? More V, oh. V is the one part of the notation that's the same. V is the concentration of transcription factors. Wait a minute. Yes, V is the concentration of transcription factors. So see, you'll notice that every line here is a function of V, okay, because the transcription model is constructed in terms of what you put into it are transcription factor concentrations along with binding affinities from sequence. What you get out is the transcription rate. Okay, and you need all the binding sites. U is the synthesis rate of message. Okay, like any other model, if you know, it's roughly like the initiation rate of message, but basically it's the rate of production. You know, it's a pleasure to talk to an audience with a lot of physicists because I stopped showing these slides in my normal talks because people's eyes glaze over. So, shall I go on? Chi is a slightly different form of this function, but it doesn't really make any difference to the actual derivation. So we derive this function in a proper way to be an Arrhenius, to be a diffusion limited Arrhenius rate. It used to look like an exponential that got cut off. So I think chi is the cutoff. Okay, I obviously have to fix this slide because the different notation causes confusion. Is everybody ready? Because I'm about to move to a coarse-grained equation. Excuse me? I've got to, I haven't published this. It's been sitting around for about 8 years. You know, it's the usual problem of the helpless professor. Because I'm not, for various reasons, Gursky's gone on to do other things, so I have to finish it myself. It needs about a week of work. And it's actually very important because it links the equations I'm about to talk about directly to sequence. So let me go on, and some of these things I think will become more obvious. Okay, I wanted to show this to this audience, okay, because the point is a lot of times you just sort of guess equations, and we do that, okay, but in this case I'm going to show the equations I'm going to be working with next as an explicit coarse graining. Okay, I might add that this is a version of the transcription model that is simpler and has fewer mechanisms than the one that I talked about in the previous lecture, okay? But you can see that calculating, if the first order term of the Taylor series involves this expression partial U of V with respect to each of the transcription factors. Okay, now I'm not going to go back to that really complicated slide, but if you remember it or you look in that Kim 13 paper, okay, you can see that to calculate this derivative by hand is essentially impossible. Okay, you could not even do this without Mathematica because those quenching terms involve a fairly insane application of the probably need symbolic manipulation programs to do this effectively. And I'm looking for a sort of mathematically sophisticated student to push this a little further.
Concentration, that's V, and that through these equations determines the rate at which the messenger RNA of the regulated gene is being produced.
Yes.
Right? And that's U of V, right?
Yes.
Now, beyond that, I don't understand what this W equation is telling us. What is it about?
Oh, I'm sorry. No. The U of V is not the. I'm very, very sorry. Let me Sanjay, I confused you. Please forgive me. In the notation of that slide, W. Actually, no, I just totally screwed up here. What an idiot I am. U is delta delta A. Okay, no wonder you were confused. I deeply apologize. This is a problem with mathematics. It's very beautiful, but typos in mathematics are extremely damaging because now, is that better? I mean, within the limits? Okay, I deeply apologize. Right. Right. And as you remember from the previous lecture, that's an insanely complicated expression. So I want to simplify it. Okay, so essentially, this partial U with respect to V is a sensitivity coefficient, okay, of the maximum sensitivity of U to changes in V. So if we gather all the terms involving V0 together and write them as H, okay, these are going to be, this is a constant because the partial is evaluated at V equals V naught. We're expanding around U of V naught. And then we have these sensitivity coefficients TA, okay? Now, I hope that, I haven't tried your patience unduly because the point of that was to derive these other equations, Okay, now historically, we were using these equations for years before we had the transcription model. Okay, and so these are the equations we use to describe the dynamics in the Blastoderm. And so what we're seeing here is this G. Okay, so here we are talking about the expression of protein A from nucleus I. Okay, as I'll show you in a moment. Okay, remember how we've been looking at things one-dimensionally along the anterior-posterior axis. Okay, we're going to index the nuclei by I, and we're going to index the genes by A. Okay, and then there's three terms here: synthesis, diffusion. Okay, now there's some diffusion happening, and first-order decay. Now, all the stuff I said about the derivation is this top term here. Okay, so look, these terms are really very basic. Okay, decay is probably more complicated than this, but you really can do quite well with first-order decay. Okay, this is just Fickian diffusion. It's like Ohm. If you haven't seen it before, it's like Ohm's law. The amount of stuff that diffuses is proportional to the concentration differences. Okay, the action is all happening up here, okay, with this same sigmoid as in the transcription model. Okay, we've flipped the axis. And the idea is that there's these various inputs here, of which the ones that occur on the left-hand side are these B sub I superscript B's through this matrix TAB. Okay, that's just these sensitivity coefficients that you could get from the transcription model. Okay, and maybe I'm spending too much time on this, but one thing that troubled me and many other people about this equation for many years is it had no connection with the actual DNA sequence. Okay, so I've basically told you how you can get that equation from something more basic. Okay, I also said something about time-dependent inputs. Okay, so it turns out when we first wrote the equations down, of course, we knew we had to have thresholds. So we put them in, and there have been various interpretations over the years, but they seem to have to do with where you take the Taylor expansion around. Okay, bicoid to a good level of approximation is constant in time, but changes with space. So the concentration of bicoid, this is a Microsoft font, that's a V, not a nu, okay, is V sub I, okay, times some coefficient m, okay, which is just like these T elements, but it affects bicoid, okay, and then there are certain inputs that do time vary, okay, Caudal varies with time, a gene that's on the two ends of the embryo, tailless, it varies with time, and the gap genes don't act on it. So these terms here do not occur on the left, they're not state variables. The TAB affects those terms that appear on the left. And one very fundamental thing about constructing, so this is essentially a continuous recurrent Hopfield neural net, if that means anything to any of you. And this is a very attractive equation, okay? People use it for a lot of things. I just want to point out that one reason to do this derivation, why I've got to get it published, is actually biology is full of situations where some enzymatic process is regulated by some complicated set of inputs and it has a maximum rate. Okay, so this suggests that generically you could use this kind of math construct for a vast variety of modeling of regulatory situations. What would make you uncomfortable about it, if I just showed it to you sort of ab initio, is any sane person would be very uncomfortable with the fact that it contains a big sum of terms. Because if you've mucked around with chemistry, you know that oh my God, it's gotten late, okay? You would know that you typically have big products and rational functions, okay? But now I've rationalized the additivity by the fact that you can do this Taylor expansion. So if I've spent too long on this detail, please forgive me. But I actually really like this, and I usually have to skip it because my usual audiences are not really interested. Oh, and one more thing, and I guess we're going to have to pause soon, okay? Oh, the computer's blinking at me. Okay, yeah, 5 minutes and I'll be at a good stopping place. So, I apologize for going so long. Maybe it's not as exciting the last thing in the afternoon as it is first thing in the morning, because I've had fewer questions. Actually, Sanjay, thank you so much for asking me those questions, because I sort of mucked up the notation there. But I just want to say one very important thing here, okay? Notice that the TABs do not have an I index, okay? That's a very important point. What that's saying is there is a copy of the same regulatory circuit in every cell. That is a basic aspect of the system that you must capture in the equations describing it, okay? And these are really now, I think of them as sensitivity coefficients. If I was telling you about this a decade ago, I would say something different, which is we don't actually know from looking at the embryos what genes regulate what other genes. And so if you don't know who's regulating who, the absolutely simplest assumption you can make is that there's the possibility that each gene regulates each other in another gene. And so T12 is simply the regulatory effect of gene 2 on gene 1. There's kind of a lot of parameters here, but not an insane number, really. Okay, one other thing about this is a big system of ODEs. Okay, because on the left, these V's are indexed by gene and nucleus. So the total number of equations you're working with can get kind of large, because the total number of simultaneous equations will be the number of genes times the number of nuclei. So, and what I'm going to tell you about, as it now turns out tomorrow afternoon, we're going to be looking at a system of 244 simultaneous ODEs. That's not as bad as it sounds, because if you were working with PDEs, you have to discretize it. My intuition a long time ago was correct. ODE existence theorems are very simple. If you're going to make a realistic model with PDEs, there's actually some math issues, because you can write down an equation and its solution may only exist for certain values of the parameters, et cetera, et cetera. And so let me close by just saying that if you have n genes and m outside inputs, you'll have n squared plus mn plus 4n parameters. What I'm going to talk about, n is 4, m is 2, so there's 40 parameters, 4,000 data points. So there's plenty of data. Excuse me? What are the outside inputs? The outside inputs are genes that affect the system that don't occur on the left-hand side. So they're typically either maternal or in some sense upstream. So bicoid is an outside input because it comes in there from the mother, okay? Caudal is in there from the mother, but it also comes from the zygote. So caudal and tailless, okay, that affects the terminal regions. They regulate the gap genes, but they're not regulated by the gap genes. So we simply represent that as a time-dependent input. And that puts us down here in the non-autonomous systems. Okay, so look, that went a little bit long. You guys are all very patient and you ask great questions. So forgive me for expounding at length. okay? Maybe, I hope it was the right thing to do. Oh, and then there's a big computational problem I won't get into in detail, which is we fit the model to data by least squares like the other model. Okay, this is actually a much more difficult fitting problem because you have to integrate the differential equations each time to get the cost function. And you really have to do this in parallel. There's actually now more data. So look, let me stop, okay? I've talked about big issues that go back to Driesch and Waddington and Tom, okay? I gave you a very puzzling problem, okay? What's happening with this really anomalous thing with the Bicoid gradient, okay? And there's something else here, okay? I'm not claiming I'm that great at doing this, but something you should look at for your own systems. You notice I obsess about some particular puzzling experimental result, okay? This is a good thing to do, okay? Because sometimes the trick is to find something that seems really, really puzzling, and it may take you like 10 years to figure it out, but a lot of times when you do, you actually learn something, okay? And there's people who are much better at it than me, but I just wanted to say that. Okay, look, it is now 5:35 in the afternoon. It's been a long day, so I'll see you tomorrow afternoon, and thank you very much. If you have questions, to the extent that Vijay decides we have time, I will answer them.
Can we go for a couple of questions? Or like everyone is tired?
I think there's plenty to be tired about. Anyway, people always find me at dinner and stuff.
Yeah, that's one thing you should always do. All right, so let's thank John once again and break.